The complement system is in humoral arm of innate immune system. Complement system, it contains over 30 proteins mainly produced by liver found in the circulating blood serum. Complement kills microbes in three different ways: opsonization, inflammation, and there is a cytolysis as well. It's a cascade system. One goes by other, ultimately it leads to exponential activation. So cascade activation complement proteins are often designated by the uppercase letter C, complement, and it's from C1 to C9. So when products are split, they become active. C1A, C1B likewise. So when there's a tissue injury or foreign body or infection, complement pathway activates in three different ways. Terms classical, alternative, and Mannose binder lectin pathway. Classical pathway is specific. It's required specific immune system, so antibody mediated. Alternative, there is no such. So in often alternative pathway, it's activated in innate immune system, but it's slow. About the classical pathway. Classical pathway is considered a part of the specific immune response. It relies on antibodies. In here, it activates C1 in the presence of antibodies. In the microbes, they are antibodies. This is C1 binding portion. So it leads to cascade activation. C1, it's in turn C2 and C4, then cleaves to form C2A, C2B, C4B, C4A likewise. It goes to the formation of C3 activation complex. It's a bridging pathway, bridging site with classical alternative pathways. Then it goes for final common pathway. So it forms C2B, C2-4B binds together to form surface and form C3 activation complex. The function of the C3 activation complex is to activate C3 proteins to form C3B and C3A. C3B is a good opsonizer. Opsonizer means molecules that bind both to bacteria and phagocytes. So it enhances phagocytosis, term opsonization. So it increases phagocytosis by 1000 folds. C3A is termed anaphylatoxins. It increases the inflammatory response by binding to mast cells and causing them to release histamine. That's why it's termed as anaphylatoxins. So once C5 activation complex is binded, it forms enough C3B. C2B, C4B make C3 activation complex has a slight affinity for C3B and C3B binds to them. When C3B binds to C2B and C4B, it forms a new complex referred to as C5 activation complex. I think these details are much not needed. If you know what is classical pathway, how it trigger, what is the C1, how it initiate, how it comes and form C3B, C2B, C5, C3A, C5A, that will be enough. So one function of C5A, it's the most powerful potent anaphylatoxin, same as C3A, but much more powerful. So other important thing is formation of membrane effects complex. Now from C5 to C9, they form MAC. Is the membrane attack complex. One bind each other and form a cylindrical structure, and they dig into the cell membrane and leads to osmolysis, cytolysis, loss of cellular content to the transmembrane channel formed by complex membrane attacks complex. That's how it forms. So it's very much important lies the capsulated as well as other organisms. Alternative pathway is part of non-specific difference. It's an alternative pathway. It's other way. So I said earlier it's a non-specific because it does not need antibodies to initiate the pathway. Alternative pathway is slower than classical pathway, but classical pathway activation antibody should be there. But in innate term, it's always activated the alternative pathway and MBL, manus binding leptin. Only difference is its initiation node. First one is C3 here, earlier C1, classical. And they are a factor B, factor D involvement. And with the presence of those, they form another C3 convertase enzyme. Here C3B, BB. Then they form, with the presence of 3B, they form C5 convertase. Same. Then go for membrane attacks complex. Alternative pathway, classical pathway, then final common pathway. So what you should know is initiate is C3 and how it initiated. Factor B is having a role here. Factor D is also another one. So with the presence of B, porphyrin, they or factor P binds to form complex to stabilize this. That's why it's mentioned here. Porphyrin plus C3B, BP. It stabilizes this complex. That's what you have to know. So then activation complex, then they lead to C5 activation complex. That's how it goes and go for final triggering or final command pathway. About the lectin pathway, activation of the lectin pathway begins when mannose binding protein MBP binds to the mannose group of microbial carbohydrate. Not all microbes activate. Salmonella may activate. This form enzyme similar to C1 of the classical component pathway that is able to cleave C4 and C2. I think you remember. Then they form C3 convertase and they go for formation of C3 and C3B, A and A, and go on. Lectin pathway trigger inflammation, same. Chemotactically attract phagocytes, anaphylatoxins, promote the attachment of antigens to phagocytes for enhanced attachment of term opsonization. Serve as a second signal for activation of naive B lymphocyte. There is a C3D. And cause lysis of gram-negative bacteria and human cells destroying foreign epitopes. This is mainly gram-negative. And remove harmful immune complex from the body by these opsonizing molecules. So now you can see all in one slide. Classical pathway, C1, MBL, mannose binding specific proteins like, then go same, here C3, ultimately they comes into the final common pathway. Enzymatic C3 convertases are different. C5 convertases also differ, but ultimately final common pathway, C5b to 9. That's lysis via O formation. So there are deficiencies. Some patients born with inherent deficiencies. So we say major pathway components. So there are deficiencies can exist from C1 to C9, maybe single, maybe multiple. So they are more susceptible for infection and delayed clearance of complexes associated with glomerular nephritis likewise. Deficiency of MBL important during infant transition to make their own antibody associated with pneumonia, sepsis, and meningococcal disease. Also, C3 deficiency most serious, key-mediated in all pathways, prone to serious life-threatening infections and immune complex diseases like infection as well as immune complex diseases. Deficiency of terminal component cause increased susceptibility Neisseria infection, including meningitis. About the regulatory component deficiency factors, hereditary angioedema results in excess cleavage of C4 and C2. Keeps classical pathway going due to this. There's an inhibitory factor deficiency. Inhibitor factor H and I produce constant turnover of C2 to deplete. Recurrent bacterial infection occur as well as due to this regulatory factor deficiency resulting edema. So you can see there are types, but what it's a hereditary disease and protein called C1 esterase inhibitor deficiency. C1 esterase inhibitor deficiency. So there is a continuous activation of classical pathway. So how we test these? So we need a clinical suspicion. So it's a immunodeficiency. So we need primary idea of this. Then we think what could be this. So rare conditions, but life-threatening conditions. So then checking that there are ways. So we do CH50. It's a classical pathway testing by hemolytic titration. There's a, then if we suspect classical pathway deficiency, we do CH50 or alternative H50. It's a ELISA test. Then we can do individual component to determine. These are costly tests. But once we need to identify, we go for initially with screening like, then go for the particular testing. Treatment, often no treatment exists. Gene therapy may be there, maybe for long-term antibiotic prophylaxis, depending, and symptomatic treatment is the best.